Today we will study certain parts of Nif genes, types of Nif genes, symbiotic nitrogen fixation, nitrogen cycles. How many studied regarding Nif gene and what is operant concept and their length and gene cluster and what is their size. So symbiotic nitrogen fixture, or as I said previously that certain species or host specific rhizobia or bacterioids can form. So these rhizobium in the root nodules of certain leguminous plant, frankie in root nodules of non-leguminous plant. Up to this, I have told you certain leguminous plant having a different kinds of rhizobia or bacteria that infect particular plant species. But now I am telling certain leguminous plant having a root nodules that having other than the rhizobia that called as plants such as alnus. Ammonia is converted to nitrates. Ammonia NH3 NH4 plus that convert to nitrates. First nitrates, then nitrates NO3 2 minus by these soil bacteria, and this process, as you know, that called as nitrification, then that is absorbed by the plant for further use. This is the general product or general phenomena without a leguminous crop. Now Nif gene organization, NIF. Two main class of Nif gene. Nif full form nitrogen fixie genes. The Nif gene and fix genes are present in rhizobia. Are present in rhizobia. The Nif gene encode NIF gene encodes certain enzymes that you have to remember. The Nif gene encode nitrogenase, and shows structural and functional resemblance. Structural and functional resemblance with the nitrogen fixing gene that present in another microbial group that is called as Klebsiella pneumoniae. Pneumoniae. Okay. Klebsiella pneumoniae and other microbial groups. Now this is all about nitrogen fixing gene. Okay. Nif gene. Nif gene encodes nitrogenase. Okay. A large number of recombination studies and transcriptome or transduction analysis have shown that the region of Nif gene, Operon, Operon, Operon concept you know, one gene, one hypothesis, and one gene, one enzyme. Likewise, Nif gene Operon is made up of 50 to 20 gene length in size. This is 50 to 20 gene length in size. Now Valentine's group. This is another scientist who have a group. Valentine groups in 1978. They have reported most Nif gene are clustered near histidine operon. First operon is Nif gene operon. Another operon, histidine river operon. So Klebsiella pneumonia, at least 20 genes linked, 20 genes linked and arranging 7 transcription unit. How many transcription unit are involved in the Klebsiella pneumonia? 7 transcription units that involved in nitrogen fixation, as well as at least 20 genes. How many genes? 20 genes. Similarly, you see here recently Hasselhoff's group, Valentine groups, then Hasselhoff groups also determine the nucleotide sequences of structural gene coding for FE protein of nitrogen fixation, nitrogenase from anabena. So this is the minimum size of the entire Nif gene cluster has been found, that is 24 kilobase. 24 kilobase means 1 kilobase means what? 1 kilobase means 1k. 1k means 1000 nucleotide pairs. 1000 nucleotide pairs. This entire region has been sequenced and they have given us many things. Now, regulation or genetics of Nif gene. How we can write in our language. Same thing, same words, same slide. I am repeating. Nothing new. Nif gene are found in both free living nitrogen fixing bacteria and its symbiotic nitrogen fixation in the various plant. Nif gene is present in free living nitrogen fixing bacteria and in symbiotic bacteria in various plant. Now, the complex of gene encoding enzyme, the complex of gene encoding enzyme involved in the fixations of atmospheric nitrogen. They are found in the nitrogen fixing bacteria that you know very well, Nitrosomonas, Nitrosococcus, and as I have already explained in the nitrogen cycle. They occur as a operon in free living anaerobic without oxygen anaerobic nitrogen fixing bacteria. What is the name of anaerobic nitrogen fixing bacteria such as Klebsiella pneumoniae? Klebsiella pneumoniae. Then Rhodospirillum rubrum. Rubrum, you can also speak like that. Rubrum. Rhodospirillum rubrum. Hmm? Then Rhodobacter capsulatus. Capsulatus. Rhodobacter capsulatus. That can also be asked in the examination. Give the exam of free living anaerobic nitrogen fixing bacteria. Now I am giving you a gene regulation. Upper in upper region you will find regulation of genetics. Genetics of what? Nif gene. So Nif gene are genes encoding enzyme involved in the fixing of atmospheric nitrogen in the form of nitrogen available to nitrogen living organisms. The primary gene enzymes encoded by the Nif gene, that is only nitrogenase complex. Remember these words. Only you have to remember these nitrogenase complex, which is the charge of converting atmosphere, which is what is the function of nitrogenous complex
Charge
is
of converting
atmospheric nitrogen to
the other nitrogen
ah.
such in ammonia.
So the primary enzyme encoded for nitrogen or Nif gene, that is complex one, and that called as nitrogenase complex. Now what is the function? Which is the charge of converting, which have a charge of converting atmospheric nitrogen to nitrogen form, nitrogen forms such as ammonia, which plant can use for various purposes. Beside the nitrogenase enzyme, Nif gene also encoded number of regulatory proteins. Now another word. Out of them, only one is nitrogenous complex is important. Third, second one is number of regulatory proteins involved in nitrogen fixation. Now, the expression of Nif gene is induced as response to low concentration of fixed nitrogen and oxygen. Now, N and O are important. So, response of low nitrogen, low concentration of fixing nitrogen and oxygen concentration, low oxygen concentration are actively maintained in root environment. Low oxygen concentration are actively maintained in root environment. Same is continuing regulation. So nitrogen fixation is regulated by Nif regulon. Now another word you have to remember, Operon and regulon. Now this Nif regulon, which is a set of seven. How many? Seven operon, which include 17 Nif genes. 770 you have to remember. Nif regulon, which is set of seven operon, which include 717. 17 Nif genes have a both positive regulator and negative regulators. Now another terms you have to remember, positive regulator and negative regulator. In most bacteria, regulation of Nif gene, transcription you know very well, DNA to mRNA. Huh. These Nif gene transcription is done by nitrogen sensitive Nif A protein. Regulatory protein I have already told you in previous slide. Nif A protein. When there is not enough fixed nitrogen available for the organism, use nitrogen C. Triggers Nif A expression and Nif A activate the rest of Nif gene. Now you have to remember Nif A, nitrogen C and Nif. Nif A and nitrogen C. Okay. If there is an abundant or you can say sufficient amount of reduced nitrogen or oxygen is present, remember the condition. If there is a sufficient amount of reduced nitrogen and NADH and oxygen is present, another protein is activated. अब दूसरा प्रोटीन एक्टिवेट होगा निफ एल. Nif L inhibits Nif A. How? Activity resulting in inhibition of nitrogenase formation. Nitrogenase formation. Non-symbiotic K. Now Klebsiella pneumoniae. Non-symbiotic bacteria carries at least 20 Nif genes, which are organized in about 8 Operon. Now 28 you have to remember and above 770. Okay. In most of the cases, Nif A means what? As I said previously, Nif genes have a both positive and negative regulator. So Nif a positive activator of transcription, and Nif L, a negative regulator. L means negative, A means positive. Okay, that controls the regulation of all Nif genes, whether it is 21, 20, or a 17. When the soil concentration of ammonia is high, NH3 or NH4. Now another condition. Nitrogen fixation is slowed down by NAFL, negative regulator. So, negatively control or gene expression by preventing NIF A to act as an activator. एक गलत करेगा तो दूसरा सही करेगा. When O2 concentration is high, now the condition you have to remember here is O2 concentration is high. Here is ammonia is high. Now this synthesis of nitrogenase decreases and leads to decrease in biological nitrogen fixation. Along with Nif gene, some of the genes also involved in several nitrogen fixations. So these are the Nif genes. You can remember A, B, C, D. Certain there are not C, A, B, D. Full ABCD is there. But you have to remember which are lacking one. Nif H means dinitrogenase reductase. H means NaH2. Then Nif D means dinitrogenase alpha subunit. Then Nif K, Klebsiella, you know very well, beta subunit of and cluster are present in Nif Y, Klebsiella pneumonia, that insert FEMO. Then Nif E, that is B2 tetramer with Nif N, and Epi, same thing as previously, but here is co-synthesis is there. Okay. Now FEMO co-synthesis, most of the things you will find in many more NIF genes. NIF V or NIF X, NIF N, NIF E, here is co-synthesis. Okay. Now nitrogenase involved in the stability of nitrogenase, that is NIF W. Then NIFL, you know very well. These two you have to remember very well. That can also be asked in the match the following. Okay. NIF-B, that is metabolic product. NIF-B co is a specific Fe and as donor to FeMO co. So ferredoxin serve as a what? Electron donor to nitrogenase. So in short, you can write FDXN. FDXN. Now NIFQ. Proposed to functions MO, then O4 processing. Okay. Pyruvate flavodoxin or pyridoxine oxidoreductase that involved in electron transport to nitrogenase, that is the NIF-J. This is all about role of nitrogen fixation and NIF genes. Now, questions how they are asked in the examination. In 12th science also they are asking same questions. In Byju's, in many platforms, those who are uploaded in net, they are utilizing same little same thing. Regulon. Regulons means what? As I said, a group of gene. Okay, that is regulated by some regulatory mode. Now, the genes of regulon shares a common regulatory element, that is binding site or a promoter site. Where it is there, that can also be asked in the examination by some scientist, or sometimes it is also asked in the NET examination, NET examination, as regulon shares a common regulatory element, then they are giving various binding sites. Okay, so you have to write binding site of promoter. Promoter. This gene comprise regulon, as I said, non-contiguously in the genome. Now, what is operon? This is a very important questions, can be asked in anywhere. In the interview also they are asking what is operon. So you have to write a unit of genetic material. Unit of genetic material that functions in the coordinated manners by the means of operator, promoter, and structural genes that are transcribed together. तीनों मिलके काम करते हैं. कौन कौन? Operator, promoter, and structural genes. Okay. That is called operon. Generally, one gene, one hypothesis we are telling. No. Okay. उसमें सब लोग मिल के काम करते हैं. So what is the functions of new genes? Now tell.
Responsible for coding of protein related and associated with fixation of atmospheric nitrogen in the form of nitrogen available to plants.
Very good. So NIF gene are responsible for. Simply you cannot write the NIF, NIF, nitrogen fixation. Okay. But you have to write Nif gene are responsible for coding of protein. Coding of protein that is related and associated with the fixation of atmospheric nitrogen. Now, in the form of nitrogen available to plant.
plants for taking that
These are
found
genes found in nitrogen
nitrogen
fixing
fixing
bacteria
bacteria and
and
cyanobacteria.
cyanobacteria, which plasmid has a Nif gene in its. So you simply write these questions. Nif is a nitrogen fixing gene and which occur in the
Rhizobium.
rhizobium. Simple. Now, which microbes is used to isolation of Nif genes? So you can write a number of hosts can be utilized for the Nif gene
Isolation.
or microbes which are used. Okay, so I am giving certain example here.
And always
Rhodospirillum rubrum, rub yani God rubrum. Rhodospirillum rubrum, uh, that is a free living and anaerobic photosynthetic bacterium, which is in addition to transcriptional control, as we have described previously, that metabolic way to this arginine residue in the nitrogenous complex. Now, where is leg hemoglobin present?
in
With
nodules.
the read.
Where is hemoglobin present?
Huh. Leg hemoglobin present.
Leg hemoglobin present. Nodules of leguminous plant.
Very good. The main functions of leguminous leg hemoglobin to facilitate oxygen supply to nitrogen fixing bacteria. Another function to protect the enzyme nitrogenase from being inactivated by the oxygen. इट मीन्स पहले वो ऑक्सीजन प्रोवाइड करता है और उतना ही प्रोवाइड करता है जितना जरूरत है. किसको जरूरत है? तो नाइट्रोजिनेस एंजाइम कॉम्प्लेक्स वही करता है सब कुछ. ठीक है. नाउ बायोकेमिकल बेसिस ऑफ लेग हिम. ओके, that is a rhizobium symbiosis. So as you know that soil bacteria, certain genera is given here, rhizobium, bradyrhizobium and azorhizobium. Azobradyrizobium. Okay. Azo and brady are collectively terms which are called as rhizobia. And they share or ability to penetrate the legume roots, as I said previously, that elicit morphological responses and that lead of appearance of nodules. I have already explained how it is enters and what the cell membranes of that entering into that, and then bacterial cell wall is dissolved and they synthesize or infection on that. So fixing atmospheric nitrogen is an immense importance in the ecological and agriculture significance. Okay. So, modern genetic analysis of these rhizobia began 20 years ago, but dozens of nodulation genes that already been taught to you that has been identified, and multiple species of rhizobia is also identified, and node factor. They are derivatives of oligolipochitins. Oligo, है ना? Here, oligochitin are synthesized by the protein products. Remember, oligochitin is synthesized by the protein product of now node ABC, node FE, and node PQ. There are certain other nodulation genes that already been taught to you. So, provoke symbiotic responses on part of the host range. So node factor together with the surface polysaccharide and secreted proteins. Remember these 7-1. Node factor together with the surface polysaccharides and secreted proteins are proposed to be the major rhizobial determinant of host specificity. I have already told you how the host is important, how they are only work on that specific host because they have a certain senses chemicals. Senses chemical means what? Node factor together with surface polysaccharide secreted protein are proposed to be a major rhizobial determinant of host specificity. So that fixing nitrogen names, as you know, that alpha, beta, proteobacters and collectively called rhizobia and symbiotic gene are required for the nodule formations, how the mitotic reactivation of corticocell leading to the nodule primordia formation and that differentiate that already been taught to you, how the oxygen sensitivity to the nitrogenase enzymes in the bacterioids that already been taught to you in the previous slide. So I will not go in depth what is cell division, how it is operating in the intermediate nodules, and certain apical a nodule meristem is present in the apical region zones that constantly generation of the new cells of the provoke continuously growth and elongated nodule shape and elongated nodule shapes. Now, what is symbiosis? Bacteria release from its present host cytoplasm and organ-like structure that is called as organ-like structure called as symbiosis. O S O N E S. Ribosomes, likewise symbiosomes. Okay, so symbiosome between the rhizobium, soil bacteria, and legume is facultative and initiated by nitrogen starvation, book starvation uh, of the host plants. So these are the questions you have to remember, and that can also be asked in the examination how symbiotic bacteria called rhizobium is important for the farmers. So you can write that root nodules of regular leguminous plant that is involved in the atmospheric nitrogen fixation, such as nitrates to nitrite, nitrite to nitrates, and ammonia. So that taken up by the soil, and then soil afterward it is absorbed by the available form of the plants. This is all about the nitrogen